tener esa felicidad de haber contribuido como miembro de una comunidad a que la gente pueda tener una vida sana y feliz.
A continuación, le cedo la palabra a Jorge Wiener, director ejecutivo de ADASA Internacional, para que nos dirija un mensaje.
¿Cuál es el significado que tiene celebrar Purim en el Día Internacional de la Mujer? ADASA fue una organización creada hace más de 110 años por mujeres visionarias, imagínense, hace 110 años, cuando el lugar de la mujer en la sociedad era marginal, no podían votar en casi ningún lugar del mundo, no tenían participación civil, tenían que pelear, y las visionarias de ADASA crearon ADASA, con una inspiración que nos acompaña a todos los que somos parte de este movimiento de ADASA hasta el día de hoy. Y cuando pienso en Purim y en ADASA, digo, ¿qué fue lo que las visionarias de ADASA crearon hace 110 años? Crearon un modelo de salvación, un modelo de cura. Y entonces me quedé pensando y digo, ¿y cuál es la historia de Purim? ¿Y qué tiene que ver la historia de Purim con la compatibilidad genética y el programa SUB de ADASA Internacional en México? Y dije, veamos la historia de Purim. En la historia de Purim es la historia de prevenir el desastre, no de traer un ejército que luche contra el desastre. Lo que hace la reina Esther en su intervención es un ejemplo que seguimos todos los que estamos involucrados en hacer mitzvot, en hacer tzedakah, en tratar de letaken et haolam, de hacer tikkun olam, de reparar el daño, de curar al herido, de curar al enfermo, pero más que nada de prevenir el desastre. Las acciones de Esther y de su tío Mordejai logran prevenir el desastre y la catástrofe y el exterminio del pueblo judío. Y es una historia mucho más grande que la historia de nuestra tribu, de nuestro pueblo, es una historia humana. Y cuando uno piensa en el programa de compatibilidad genética, es la historia de Purim. Es la historia no de tener que curar al enfermo, sino de prevenir que el enfermo aparezca. No de tener que arreglar lo que está roto, sino prevenir que tengamos que hacer eso. Es prácticamente un milagro. Y va a haber un milagro que va a suceder. Cuando se abra dentro de 15 años, va a haber jóvenes que nacieron en estos años, que nacieron sanos, por algo que ADASA y la Comunidad Judía de México hicimos juntos. Y valió la pena que pusiéramos eso en la bóveda, para que cuando lo abramos podamos marcar ese momento tan importante que México y ADASA juntos crearon algo para México, para la salud de esta comunidad, pero también un ejemplo para el mundo, para que podamos seguir reparando el mundo, previniendo, curando e innovando.
Porque darías la vida por ellos, prevenir está en tus manos. Infórmate.
Hola, yo soy Teddy Husni y ella es mi esposa Mijal Tawil. Ambos pertenecemos a la comunidad Maguen David y yo me considero un caso de éxito del estudio de genética. Nosotros nos enteramos de este estudio por mera casualidad y gracias a Dios nos puso en el camino este estudio.
No queríamos hacérnoslos porque no queríamos recibir una mala noticia y no entendíamos por qué hacérnoslos. No teníamos ningún antecedente en nuestra familia que nos hiciera pensar que necesitábamos hacernos este estudio o que tuviéramos una condición que pudiera hacernos propensos a tener un hijo con una enfermedad genética.
Me acuerdo perfecto el día que nos metimos al Zoom con la doctora Orly, cuando nos dijo, ustedes tienen una incompatibilidad genética. Dije, no puede ser. O sea, ¿qué es eso?
¿Por qué?
¿Qué va a pasar? Y a los 15 segundos nos sentimos bendecidos. Dijimos, espérate, nos está dando la mejor noticia de nuestras vidas. Porque gracias a esa noticia vamos a evitar una enfermedad que luego nos enteramos que era terrible, o sea, devastadora.
Las probabilidades de que tuviéramos un hijo con esta enfermedad es del 25%, es decir, 1 de cada 4. Es una probabilidad que me parece altísima. Una de las dudas más importantes que teníamos era qué hacer con el resultado, ¿no? O sea, ¿yo para qué me quiero enterar de que puedo tener un hijo con una enfermedad, ¿no? Hoy entendemos perfectamente que no es para qué quiero saber. La información es información que te da muchísimo poder para tomar una decisión en la que evitas al 100% tener un hijo con una enfermedad genética.
No es yo para qué quiero saber. Nuestros genes ya están adentro de nosotros. Nos den la noticia o no nos den, nuestros genes no van a cambiar. Pero si nos dan la noticia, podemos evitar el problema.
Porque hay una alternativa en la medicina, que es la fecundación in vitro, en donde en parte del proceso pueden elegir a los embriones que no tienen las dos copias del gen y implantar un bebé, un embrión completamente sano.
Me permito presentarles a la doctora Agar Morshaket, genetista y jefe de bioinformática del Hospital Hadassah de Jerusalén, para que nos presente los resultados que han encontrado en este estudio tan importante para nuestra comunidad.
And my expertise is analyzing genetic data. I'm super excited to be here today and to tell you about this awesome project, the exome-based screening of the Jewish community in Mexico. So, as I'm going to walk you through some basic genetic introduction, but before I do that, I just want to talk for a second about the essence of genetics. As people in the present, we all have past and we have future. Our past defines who we are as persons with heritage, with history, with ancestors, with habits, with families. The decision that we took in the past and that we take in the present defines our future. In Hebrew we say, Da, me'ayin bata ve'le'an ata holech. This is, we say, it's not we say, it's from Mishnah Masechet Avot. And what it means is that you should know where you come from and where you are going. In this talk, I will try to present where we come from as a one community and where should we go. So as Jewish, as a community that avoid assimilation, hitbolelut, who lives in one isolated community, we all share genetic traits, common genetic traits. And numerous publications showed before that if we take individual and we test them for their genetic composition, the genetic variants that they hold, we can segregate Jews and non-Jews very easily based on a simple genetic analysis. Well, this is fascinating. Our DNA binds us together. But what's DNA at all? So let's just try to explore this for a second. DNA is a molecule that stores the instructions of our body's work. It is passed on from parents to their offspring, parents to from generation to generation. And it dictates the way we behave, the way we look, the way we're healthy or unhealthy, affected. It's a diary that contains fascinating stories that starts from our ancestors through history. And about 100 years ago, there was an immigration from Syria to Mexico. About 20,000 individuals immigrated. You probably know this much better than I do, but from the genetic point of view, this is a fascinating phenomenon. Because when a specific population immigrates from one place to another, the new community has changed or might have changed the genetic diversity it holds. And now there is a new community that starts over and inbreed. And we need to understand what are the impacts, what are the influences of this community in order to take decisions to the future. So we said DNA. The DNA is composed of 4 letters, a simple language, A, C, T, and G, very basic language. The DNA is found in every nucleus that is found in every cell, which is the basic structure of our bodies. So our body is composed of trillions of cells. Each cell contains a nucleus, each nucleus contains a DNA. Well, DNA is not just found there all over. It is condensed. It is condensed inside chromosomes. You see, these are the chromosomes, and this is the DNA. The DNA is the language that instructs how our body is going to work. So we said chromosomes. We have 23 pairs of chromosomes. One we receive from our mother and one we receive from our father. And this is a very important point here. 22 pairs of chromosomes we all share, 1 to 22. Again, one from our father, one from our mother. The last pair of chromosomes is the pair that dictates the gender. So XX for females and XY for males. This is all what differentiates males and females. So now we better understand the genetic composition that we are, right? We're half our father, half our mother. When we talk about family tree, when we talk about genetics and we want to illustrate the past and the future, we can describe it like that, you know, with a family. You're all familiar with this kind of illustration. The present would be, for example, this person, with the past being the ancestors, the fathers, the parents. This is just one generation, but it goes way back. And the future are our children. This is the future that we build. This is how our genes are going through one generation to another. As geneticists, we like to put things complicated, so we put it like this, and we call it a pedigree. In this pedigree, the square is a male, the circle is a female, and this is how we describe a family. If we just take a look for a second on the genetic composition by colors, we would see that, for example, this person here, this individual, carries half of the mother and half of the father. And if we look one generation below, then it's already a quarter from each of our grandparents. And this is how it goes. Of course, one can also paint it like this with gummy bears, but this would be less scientific. So let's talk about genes. Genes are the instructions of the body to make proteins, to code for proteins. They're the most basic instructions. It's like a recipe. The body needs to know how to work. The language is written inside the genes by the DNA. And now we're going to dive into more terms of genetics that are important for this talk. A variant, a very important term. A variant is a change in the DNA between one human genome to another, to a reference genome, for example. A variant can be homozygous when the two copies of the gene has the same variant, which means, again, I'm going to repeat it a lot today, one from the mother, one from the father. If I have a variant in the two copies, then I'm homozygous for this variant. But a variant can also be heterozygous, what we call a carrier. So that means that one copy is normal. I would say normal, but it doesn't mean that it's not normal to have the variant. It just means that it's like the reference, and the other copy carries a specific variant. And it's important to understand, variants can be benign or they can be pathogenic. The benign variants that we carry are the ones that differentiate us from one another. We do not think the same, we do not behave the same, we do not look the same. Exactly for these reasons, we all carry variants. We all are carriers for variants. But a variant could also be pathogenic. Pathogenic means disease-causing, harmful, and if a specific person is a carrier of a pathogenic variant, he might be affected. A genotype, I have two more terms. A genotype is a set of a specific variants in a genetic specific region of the human genome. So this is if we look on a specific gene, we read the whole sequence, we take some variants and we say, this is the genotype of this person. The phenotype on the other side is what we see, is the observed characteristics of that genotype. So there is the genotype that is found in the DNA, and there is the phenotype, which is what we see. It could be the color of my eyes, the color of my hair, it could be if I'm affected or I'm healthy. So all of what we see from the outside is the phenotype. And genetic variants are inherited from one generation to another in two main genetic models. The first one is dominant and the second one is recessive. And these inheritance models are very important here to understand. And all that I said so far was just to explain this slide. So let's see if you're with me. A dominant trait would be a trait that is expressed in the heterozygous state, which means it's enough that I have one damaged copy of the DNA, one variant, a single variant, not two, and a disease will be caused. That's a dominant inheritance model. There is 50% chance of inheriting each of the different copies of the DNA, which means, for example, if one of the parents have a specific dominant condition, then each of their children will have 50% chance of having the same genetic disease. But from the other side, we have the second inheritance model, which is recessive. Recessive means that in order for us to see a phenotype, in order for the genotype to cause a phenotype, the patient should be homozygous, meaning the two copies of the DNA should be damaged in order for us to see the phenotype, like this. Well, this happens when both couples are carriers. So both parents should be heterozygous, like here. And then there are 50% chance of inheriting, for each parent inheriting the damaging copy, the damaged copy. And now, this is just basic statistics. In order for this couple to have an affected child with a homozygous variant, if both of them are carriers, is 25%, because we need this copy, damaged copy, and this damaged copy. So it's this and this. That means that if two persons carry a pathogenic variant in the same gene, it doesn't need to be the same variant exactly, then every child, and the chances are per child, they have 25% to be affected. For this reason, there are about 1%, and this is from a known publication, there is about 1% of all couples around the world. This was done on European couples, but it's relevant for all couples to have shared carrier status for a pathogenic variant. So this is just worldwide statistics, okay? 1% for a couple to share a common genetic variant in the same gene. In first cousins, in couples that are consanguineous, that are related, the chances go much higher. Well, this makes sense, right? If I am related to you, we share more of our genetic composition, more likely to share the same variant in the same gene. So it is actually 16 times higher than regular couples that we just that are not related. But the question here is, what about isolated communities? They're kind of in between, right? Because an isolated community, of course we understand, they have the increased risk of having shared genetic composition traits. Well, that's clear. How much higher? This is a question that we tried to address using this program. And I just want to say, the smaller the community and the more isolated it is, then the chances are higher. And again, this is just basic logic. This is not science. This is just saying, if I'm related to you, if we come from this, if we have the same founders, we are sharing the same genetic composition. Well, this holds a great advantage in terms of community, in terms of being together. This also holds risks for having affected children. And let's just for a second talk about what are we talking about? We're talking about rare diseases. I hear that a lot, but that's rare, right? Why should I be worried? It's rare. Well, yes, it's rare, but every disease is rare, but altogether, they're pretty common. So we know today that there are 300 million of patients around the world suffering from various different rare diseases. So the rare is not that rare. And actually, more and more companies in high-tech and startup companies are starting to work on rare diseases because they understand someone needs to tackle this. We cannot say it's rare. There are only 20 patients around the world. This is not good enough. We need to provide a solution, and that's becoming very clear. But because the rare genetics are not that rare, and because the chances of having a shared genetic variant in the same gene is not that rare, a whole field was evolved during the last decade. This is called preventive genetics. And when we say preventive, we mean that we want to offer couples at risk a way to minimize their risks by performing a preconception screening test. In many countries, such as Israel, there are governmental programs dedicated for this. So if a country acknowledges the risks that is involved in having children, it offers the pre-screening tests, and this minimizes dramatically the risk of having affected children. In Israel, many diseases completely disappear. Tay-Sachs, and of course this is a lot to do with Dor Yesharim, that started from America and came also to Israel for the more Orthodox community, and it completely prevented specific diseases from the hospitals, from the families. So it's well proven that preventive genetics is beneficial. Now, there are two options. There are two options to do preventive genetics. The first one is to do a gene panel or variant panel, which would be relevant to your ethnicity. So in Israel, you would go to the clinic, they would ask you, what is your ethnicity, what is your ethnical background, where does your grandparents comes from? Okay, you need this list of variants to test, which is a good approach when you already sequence the whole population and you know what's common and you have already recognized and detected all the common diseases, all the founder effects that a specific population have. Well, that's not really the case in many different communities. And for this, there is the second approach, which is exome sequencing. Exome sequencing means let's sequence everything. Let's not just concentrate on what we think is prevalent and what's common and on what is severe. I want to sequence everything and I want to learn. I want to understand what's prevalent and not prevalent. I want to characterize the specific ethnical background that I'm looking at. So exome sequencing, just a few words. Exome sequencing is the reading, the sequencing of all the coding region in the human genome. Surprisingly, this is only 1% of the human genome, so it's actually sequencing a small part of the human genome in order to have a lot of information, because when we say coding regions, we mean all the genes that are coding for proteins, and usually diseases occurs when variants are found in these specific genes. So cost beneficial, it's a great approach. We sequence really a small portion of the human genome, but we receive a lot of information to handle. And the exome sequencing sequencing is one of a kind for each person. So I can take the test now, it will be relevant for 20, for 30, for my whole life. This would still be relevant. And every person is one of a kind. Like a QR code or a barcode, unless you have an identical twin, you are the only one to have this exome signature that we are testing. The use of exome sequencing in preconception carrier screening is well known, and there are thousands of publications describing this. I just picked 4. One of them is ours. We published, I think 2 years ago already, a publication about exome-based carrier screening. But as you can see, there are many others. By now, it is well accepted that closely related couples, consanguineous couples, this would be the best approach. I mean, this is not an argument anymore. This is not open for discussion. This is the scientific clinical recommendation that a couple will receive if they're going to a clinic that can offer an exome sequencing. Because for a consanguineous family, it is not important what the founder have, what the ethnical background have. They have their own family variants that needs to be tested. But let's just talk for a second about what are we reporting when we do an exome sequencing. So of course, there are tons of labs around the world, and we need one structured way, standardized, to report variants. So we follow, like every other lab, genetic lab, the ACMG guidelines. The ACMG is the American College of Medical Genomics and Genetics, and they are a committee that is dedicated to provide guidelines. They classified variants into 5 categories: benign, likely benign, variant of unknown significance, likely pathogenic, and pathogenic. Well, it's kind of clear that the benign and likely benign are not something that we are going to report when we do a carrier screening. Let's talk for a second about the other 3. I just want to say I'm going to use these abbreviations. So we say VUS, it's already a common word in English. So when we say VUS, we mean variant of unknown significance. LP is for likely pathogenic, and P is for pathogenic. And we treat likely pathogenic and pathogenic the same. So for me, it's just pathogenic. So when we do exome compatibility test, we report on 3 things. The first one is shared carrier status. And now I'm talking about this program that we built here together. We are reporting likely pathogenic variants of both couples, and also when one of the couple is a carrier of a VUS variant of unknown significance, and the other one is a carrier of a pathogenic variant. That is because we see 50% chance we already have one pathogenic variant. If we see a rare, uncommon variant on the other side, meaning on the other individual in the couple, then we're kind of worried that we might miss something. But when we report a VUS, just again a variant of unknown significance, it means that we just don't know. This is important to understand. We are limited as scientists. We know what we know. We learn. We sequence, we learn. We investigate, we learn, but we are limited. In addition to the carrier, the shared carrier status, which is the essence of this test, we also report on X-linked maternal pathogenic variants that are passed in 50% to each of the offspring, and we report secondary findings, which are medically actionable variants that could save life. This information could save life. For me, this is the benefit of an exome sequencing. I can decide what to look at. I can decide to explore secondary findings, and I can decide to ignore them. I have the ability to choose. So this is one very important point here. We can opt out. The couples can opt out from being reported VUS variants and also secondary findings. The other important thing is that this test could be customized to the community. So this is work in progress. We work, we started, we review it every now and then, we stop, we analyze the data, and we decide. We take decisions together. This is a collaboration in which we work together. And when we say it could be customized, it means you can decide about the timing. You can tell, okay, I want to have the report before the shidduch. I don't want to have it after. That's fine, we can do it. It's only a matter of when you take the test and when we report. We can handle this. You can tell me, I don't want to have VUS, I don't want to have even likely pathogenic. I only want to have a strict list of variants, and I want to have it from the exome sequencing, because then cost effectiveness, it's much cheaper than do a lot of other broad tests. So the exome test is a very wide one, but we can close filters, and we do that day by day. We create protocols, and when I close the filter, I don't see anything else. So I'm protected. I don't need to see information that I can't report, and you receive what you want to receive. Every genetic test has limitation. If we don't understand the limitations, if we are not here today to clarify them, for me, I did nothing. This is the most important slide. Because when you do a genetic test, it's important for you to understand what you receive and what you don't receive. So what you don't receive here is, for example, Fragile X syndrome, which is one of the common syndromes of mental retardation that are not covered by the exome sequencing because the variant is of a different kind. It is a repeat expansion variant, meaning a 3-nucleotide unit amplifies 200, 300 times. The exome cannot sequence it because it just keeps confusing with counting the repeats. And this is a tough limitation. When you do exome-based panel or exome-based screening, or a panel, because it's the same, you have to do Fragile X in addition. Oh. God. Okay, sorry. We have coverage limitations. So there are 20,000 genes in the human genome. We sequence them all. But once upon a time, a small portion, could be 1%, could be 2%, of the exome regions are not covered. This is an error rate of the method. Every method has an error rate. It's very important to acknowledge that this might happen. We are also limited with variant interpretation. I think this is the most important point here. When we see variant of unknown significance, it doesn't mean that it's not pathogenic. It also doesn't mean that it's not benign. It just means that we don't know. And we don't know when we don't have enough healthy individuals sequenced. When we have a lot of background, a lot of reference, then it's easy to compare. I say, okay, I've seen this variant before in a healthy individual, I'm good. If I don't have who to compare to, if I don't have a database, then these are when the VUS variants become disturbing. And this is exactly the situation that we're facing right now. The exome is also limited to detect structural variants, which is a different type of variance that is not read by sequencing. And I think also very important to mention here, de novo variants. So de novo variants are variants that occur in the child, not in the parents. They are not inherited from the parents to the child. Each and every one of you here, and myself included, carry between 4 to 10 de novo variants. This is the small contribution of each generation to the other. These de novo variants in our ancestors, these pathogenic ones in recessive genes, were exactly the variants that we are now facing and we are trying to avoid. But a de novo variant could be anywhere. It could be benign, it could be pathogenic. It can also cause a severe dominant disorder. This is something that we don't test, as we don't even have a sample of the fetus of the child to be. So we can do carrier screening, we can do it all day, we can do genomes, we can do the broader that we want. We cannot detect what the fetus has as a de novo. So the zoo compatibility test, and I want to show you some results, okay? The zoo compatibility test started 3 years ago. I was starting to analyze the data from day one, and we have so far about 433 couples that were already analyzed and summarized, and we have dozens under processing right now. And this is the main results of this presentation. About 10% of the couples received a positive result for shared carrier status. This includes couples of likely pathogenic and likely pathogenic, with pathogenic and also VUS with pathogenic. So we included both groups here and we got to 10%, which is very, very high. If we only look on pathogenic variants for a second, we see that it's 3%. So for those that are actually listening to me right now, in the general population there is 1%. This means that the Jewish Mexican community has 3 times higher risk than a regular unrelated couple if we go to, let's say, European couples. So yes, this is the conclusion that we can draw here very clear. Among the 13 positive couples for, again, only pathogenic variants, these were all couples that were referred to IVF and PGD and procedures, etc. 8 of the couples had exactly the same variant, meaning that their offspring to be, like their child, could have been homozygous for their carrier status, shared carrier status. 5 of them were carriers of different variants, but on the same gene. This is called compound heterozygous, like this here. What that means, or that implies, that more than half of the couples had exactly the same variant. This is an evidence that couples inside the community share their genetic composition. So there is a high sharing of genetic traits inside the community. The 13 couples received shared carrier status in 11 different genes. So we are not talking about a specific variant, a foundry variant that is found in many of the couples. We are talking about different types of variants, different diseases, which is, for me, was fascinating. And what's very important to understand that if these couples were to do a panel-based approach, only 5 of them would have been detected. That means that 6 out of these couples would receive a negative result, reassuring them that they are good to go, but actually they had a severe disease, 25% chances to have a child affected with a severe disease. This is why we think that exome sequencing is the best approach for this community. And now if we take all the individuals, and then now I'm not putting them into couples, I'm just looking at all the individuals all together, and we just say, let's look on the pathogenic variants. Because, you know, a couple is just 2 people that met, but I could create couples randomly from the scientific point of view. What we saw was very interesting. 2% of the community, of the individuals that were tested, were positive for a pathogenic variant in COL6A2. 2%, that's extremely high. When we go to GNOMED, which is the largest publicly available database that we work with in order to compare the data that we have, in GNOMED there are more than 5,000 Ashkenazi Jews, and these cohort of Ashkenazi Jews are very helpful for us when we do analysis for Ashkenazi Jews. But when we go there, it gives us 0. So while it's very common in the community, it's completely absent from GNOMED. And there are more examples to show that the Mexican Jewish community carries unique signature variants that are not found in other populations. And just to provide you a basic explanation of the types of diseases that we are reporting, because it is also very important. So I will provide only 2 examples because this is exhausting and not very pleasant, but the COL6A2 that I just mentioned is a cause of Ulrich congenital muscular dystrophy. This condition, while it does not affect the intelligence, it causes a severe congenital myotonic dystrophy that deteriorates a long time. And while most of the patients are able to walk, after a significant time they lose ambulation and they lose the ability to, and they have a decline in respiratory function by the age of 10 years old. So this is a severe disease. Another disease that was found and reported was Cohen syndrome. How funny, Cohen syndrome. That was VPS13B gene, which was found pretty prevalent in the community. And this is a cause of a severe disorder of microcephaly, which is a small head, mental retardation, intellectual disability, global developmental delay, recurrent infections, and a lot of other dysmorphisms and specific features of the syndrome that are pretty common. The variant is pretty common inside the community. So we have the base to know, the base to think that couples should continue in doing exome sequencing. And why should the couples do exome sequencing? So I thought I'm going to be the first one to say today, but I think you already received, I don't understand Spanish, but from what I understood, we already got an explanation about IVF and PGD. But really shortly, knowledge is power. We use that a lot as geneticists. We say knowledge is power. If you know, you can act. You can define your future. You can design it. You can affect it. So the couples can have procedures of IVF and PGD, which is in vitro fertilization and pre-implantation genetic diagnosis. A lot of confusing words to say. We can create embryos in the lab, we can grow them for a few days, take one cell, test it for the specific family mutation, and then differentiate between healthy fetuses, affected fetuses. We throw the affected fetuses to the garbage, literally to the garbage. We take the healthy ones and we can put them back in the womb and offer the family a healthy offspring, a healthy child. So again, knowledge is power. This is not an information that I give you because it's fun. This is information that is important for the health of your future. Secondary findings, though. I see that I'm skipping slides. Okay, so just 2 slides on secondary findings. One will be an introduction, one would be the results. So secondary findings are kind of adverse effect or something that we see in the data that we don't want to ignore. The ACMG, same ACMG as before, has defined a list of 78 genes in which they say, if you find a pathogenic variant in this list of genes, you should report it because the reporting will be actionable. Again, when we report, knowledge is power. We do not report because the knowledge is important. We report because there are actions to take. For example, if a specific person is a carrier of a cardiomyopathy, then checkups, then there are a lot of cardiology tests that we can refer the patient. These can be life-saving, and this is why they're so important. And there is an option to opt out. We highly recommend not to do that. This is the benefit of the exome sequencing. I'm not limited for a specific panel. I can provide life-saving information. It could be prevention. For example, if a specific woman is a carrier of BRCA, we can have specific protocols to prevent 0% versus 85% of having breast cancer. This is a game changer for us. This is super important. And even if we don't prevent, we provide early detection, we delay the onset. So this is the results of the individuals that we tested so far. We found 34 individuals were positive for one of the genes of the ACMG secondary findings. This is almost 4%. Then we always compare to, public publications of the general populations. They all state about 2% ACMG secondary finding positive rates. So almost double. The reason for that is that there are specific founder variants in these lists of genes that are causable. For example, the BRCA. We all know that in Ashkenazi Jews, there is a high risk of a founder effect for BRCA mutations, which of course affects this information, this data. So the 34 positive were positive for 22 different variants and in 14 different genes that were recurrent. And this is the distribution of variants among the genes. I think I haven't said it, but the list of 78 genes are all cardiovascular and cancer. So both of these fields are actionable, are preventive. And if we look for a second on BRCA2, we see that we have 6 women who had a positive or had a pathogenic variant in this gene. We can offer a protocol. There is a clear protocol for carriers that could completely prevent disease. This is almost the last slide. As scientists, we always ask ourselves, how can we improve? We want to be improved, we want to be better, we want to learn from the data we are analyzing, we're improving. If you don't do that, something is wrong. So we can improve in 2 ways here. One, the basic way to improve is to stratify variant of unknown significance. Because if I have a couple that both are carriers of a VUS variant, and I'm not reporting it, it still might be pathogenic. If I want to report, I have 2 options here. The first one is to test affected individuals from the community. So best practice in genetics, always start from the affected individuals. When I do carrier screening, I look on a couple and I hypothesize the fetus to be. I don't have the fetus, I don't have clinical presentation. I don't know what it can carry or not. But when I have an affected child and I have a clinical presentation of, let's say, blindness, now it's much easier. VUS variants become pathogenic. This is the way to stratify VUS variants. We need to test affected individual from the community. And if we do that, and if we do find variants that are more common than we think, we can go back to all the couples that were tested and inform them of their risk. The other option, or the second option, and I think we should take both of them, is to combine clinical data together with the genetic data. So we're kind of limited at the moment, right? We say ACMG secondary findings, but I don't have the EKG, the cardiograms of all these patients. If I had it, I would be much more confident to say this is a VUS, this is pathogenic, this is benign. We need to start to combine clinical data together with genetic data. And when I say clinical data, I mean a thorough clinical data. This is, for me, it's the phase 2 of this program, and it's on the way. With that, I would like to end, to finish. I have a long list of thank yous, so please be patient here with me. First of all, I want to say thank you to Professor Orly Pelek. She was the one to initiate this program 3 years ago. She came back to Israel and she told me, Hagar, we need to do it. Start analyzing the data, start designing this project. And this is what we do. I want to thank to Professor Tamar Ariel, who is now the current managing, or the head of the genetic department at Hadassah. I want to thank all the clinical department and all my great team that is doing this work, amazing work. Dr. Rivka Birenbaum, who is doing most of the analysis currently, and Sarah Weissman, who is handling most of the samples and making sure all couples arrive, written properly, and we produce a report properly. This is the team. I want to thank the Zug committee. Sassy, thank you. You are amazing. I mean, without you, there's all your support in this project is amazing. I want to tell you something. You're doing not less than avodat kodesh. This is what it is. And I'm amazed, we are at Hadassah, we are amazed with the actions that this community had taken during this last 3 years. And this is exactly da me'ain bata ve'le'an ata holech. Thank you.
Al conocer a la doctora Orly, su primera pregunta fue: ¿se realizaron algún panel de compatibilidad genética antes del embarazo? Y claro que no teníamos idea de lo que nos estaba preguntando. Informándonos, aprendimos que en ese momento existían muchos paneles diferentes de compatibilidad genética. Pero aparte de que en México no se acostumbraba a hacerlos, ninguno de ellos hubiera detectado el defecto genético que portábamos Eduardo y yo, y de igual forma no hubiéramos podido prevenirlo. Entonces, es así como surge la necesidad de crear un proyecto en el que las parejas de la comunidad pudieran tener la oportunidad de sacarse un exoma completo en modo de compatibilidad genética. Esto no existía en ningún lado. ¿Para qué el exoma completo? Era una locura, no era necesario, muchos decían. Y me atrevo a decir que muchos lo siguen diciendo. Pero hoy, muchos de los aquí presentes estamos conscientes de la importancia de este estudio y del privilegio que tenemos para poder llevarlo a cabo. Me gustaría agregar que nos hemos topado en bodas, eventos sociales, con diferentes parejas que han tenido un resultado positivo y se acercan con nosotros y nos dan las gracias, ¿no? Y la verdad es que nosotros solamente fuimos dos valientes que nos atrevimos a contar nuestra historia. Pero es gracias a los chavos, que aparte me gustaría agregar que ojalá que pronto estas juntas estén llenas de chavos. Porque es muy respetable el público aquí presente, pero no hay muchos chavos. Entonces, que los podamos llenar estos foros de chavos y que los sigan haciendo, porque no es gracias a nosotros, sino es gracias a ustedes por tener el valor de hacerlo y de que esto siga teniendo continuidad para que podamos todos seguir teniendo bebés sanos. Y bueno, para terminar, me gustaría nada más decir que pues, cuando vamos a tener un hijo, siempre sabemos que nos va a cambiar la vida. Y en ese momento nosotros sabíamos que iba a cambiar la nuestra, pero nunca nos imaginamos que aparte de transformar nuestras vidas, íbamos a traer al mundo a una personita que cambiaría la vida de tantos por el simple hecho de existir. Gracias a todos por estar aquí y gracias por seguir ayudándonos, a seguir haciendo función. Muchas gracias.